Hey guys, this is our first digestive lecture video. We're going to be focusing on the small intestine. In previous lectures, we talked about everything before the small intestine, but here we're going to be talking about small intestine and after. So with the small intestine, this is where mostly all of the chemical digestion and nutrient absorption occurs. So if you remember from previous lectures, we had some digestion occurring within the mouth, like with enzymes such as salivary amylase, or within the stomach. However, most of it's going to occur within the small intestine. It's important to note that with this video, I'm only going to cover the very basic points that need elaboration, I think. So it's up to you to read the book and read this entire lecture. So the small intestine is the longest part of the digestive tract, with small referring to the diameter rather than the length. So this is the anatomy of the small intestine. If you remember, the small intestine begins at the pyloric valve, with the duodenum happening right here. So the duodenum is the first part, and of course this is the stomach. So duodenum, and then the jejunum, and then the ileum. It's important to note that the ileum here is I-L-E-U-M. You might have seen a word like this before when talking about the pelvic girdle or pelvic bones, where you have the ilium, the ischium, and the pubis, but that's I-L-I-U-M. So you need to spell I-L-E-U-M when you're referring to this, or else it would be wrong on any tests in this class. So like I said, the small intestine is divided into the three regions of the duodenum, jejunum, and the ileum. So the duodenum is the first approximately 10 inches, and it begins at the pyloric valve, and this is where it receives the major and minor pancreatic ducts. So remember, we get all the pancreatic juices being going into there. So the duodenum receives the stomach contents, which at this point is called chyme, as well as pancreatic juice and bile. The stomach acid is also going to be neutralized there. So if you remember, chyme is pretty acidic as it's coming from the stomach, so you have to neutralize it or else it's going to start to erode the duodenum, which is not a very good thing. Fats are physically broken up, which is called emulsification, or they're being emulsified by bile acids. Pepsin, if you remember, that's one of the enzymes within the stomach that breaks up proteins. That's inactivated by the increased pH of the small intestine as the stomach acid's being neutralized. And then it's important to note that the pancreatic enzymes themselves are what's going to perform the chemical digestion within the duodenum. So they're not actually there's not actually small intestine enzymes, but rather the pancreatic enzymes. But we'll talk about that in a second. So the second part of the small intestine is the jejunum. And this is relatively thick and muscular, and that's how they notice, or that's how they denote that the jejunum versus the duodenum, because it's not like it's marked when you go inside of a body. It doesn't have a label that says jejunum. Most of the digestion and nutrient absorption itself occurs within the jejunum, specifically of the small intestine. Okay? And then we have the ileum, which is the last part of the small intestine. This is thinner, less muscular, less vascular, and because of that, it's going to be paler pink. It ends with the ileocecal junction, which is the small intestine, where the ileum is going to join the cecum of the large intestine. And of course, this also has a sphincter, which is called the ileocecal valve, which is formed by the thickened muscularis, which is one of the layers of the small intestine. And it's going to protrude into the cecum, which is the very beginning of the large intestine. And this functions to regulate the food passage into the large intestine so it doesn't all just kind of squirt out at once. So for circulation, the small intestine receives most of the blood from the superior mesenteric artery, and the blood leaves with the superior mesenteric vein, where it then joins the splenic vein and goes to the hepatic portal system, headed for the nutrients, or headed for the liver with all the nutrients that it just absorbed from from the food within the small intestine. And of course, this is important because it's going right to the liver so that it can be metabolized or detoxed if there's anything that needs to be metabolized or any toxins within it. Okay. So the small intestine is a really good example of where form meets function because the small intestine tissues are designed, if you want to use that word, for nutrient digestion and absorption. So because of that, there's the layers aid in peristalsis, which is that movement of trying to move it from the proximal portion, which is the closed portion, to the distal portion or the far portion of the small intestine. So it moves out so it can eventually be defecated. And it also has a very large internal surface area, which helps to aid with digestion and absorption. So there are three main ways that it increases intestinal surface area, and these are the circular folds, the villi, and the microvilli. So circular folds are the largest folds of the intestinal wall and involve just the mucosa and the submucosa. And these occur from the duodenum to the middle of the ileum, but not the furthest part of the ileum because it doesn't really matter at the far part of the ileum because pretty much everything should already be absorbed or digested. So these circular folds cause chyme to flow in a spiral path so that it causes more contact with the mucosa itself, and this promotes more thorough mixing and nutrient absorption because you want you really want you want the thorough mixing of the chyme so you can really digest it a little bit better and also absorb all the nutrients a little bit better. So villi are finger-like projections, and which you can see here. And these have epithelial cells that are joined by tight junctions that are going to prevent the digestive enzymes from seeping between them, which, as you can imagine, would be pretty bad. So each villi itself, each villus itself, contains an arterial capillaries, venule, and a lymphatic capillary, which is called a lacteal. If you remember, here's the lacteal right there. Remember from our lymphatic, the lacteal functions in lipid absorption. So microvilli form a fuzzy brush border on the apical surface, which is like the outside surface of each absorptive cell, so each surface or each cell that absorbs nutrients. And this, of course, increases the surface area for it to absorb. And they also secrete brush border enzymes, which are in the plasma membrane of these microvilli, and they carry out the final stages of enzymatic digestion. These enzymes are not released into the lumen, so that means they don't go into like the circular, the main part of the small intestine. And so because of this, it's a very important term. This is called contact digestion. They carry out contact digestion. And so the chyme, which is the food mixture, needs to come in contact with the brush border in order for digestion to occur. So the intestinal churning of the chyme, which is mostly from the circular folds of the small intestine, means that there will be contact with the mucosa so that it can carry out the contact digestion. Okay? So there are also intestinal crypts, which are little pores that open into tubular glands on the floor of the small intestine between the bases of the villi. And these are kind of like the gastric glands that we talked about. So what I want you to know for this is that the few cells that are clustered at the base of each crypt, and these are going to function in, these are defensive proteins that help resist bacterial invasion of the mucosa. So these secrete lysozyme, phospholipase, and defensins, which again help resist bacterial invasion. So remember, intestinal crypts help with resistance of bacterial invasion. So also within the duodenum, there are duodenal glands which secrete bicarbonate-rich mucus. Bicarbonate-rich mucus functions to neutralize the stomach acid and shield the mucosa from the stomach acid's erosive effects. So remember, we talked about how important it is that you neutralize the stomach acid so it doesn't erode the duodenum. And this also aids in inactivating pepsin. So these intestinal crypts, remember those are where the bacteria, they help with bacteria. They also secrete intestinal juice, and this is in response to acid, hypertonic chyme, and the distension of the intestines. And these intestinal crypts, the intestinal juice is made up of water, mucus, and some enzymes. Most of the enzymes that function in the small intestine come from the brush border, which again, remember those are the microvilli, as well as pancreatic juice. So the contractions of the small intestine we talked about a little bit. So they have three main functions. One is to mix the chyme with the intestinal juice, bile, and pancreatic juice, and this digests nutrients more effectively and neutralizes the acid. It also helps to churn the chyme to bring it into contact with the brush border enzymes, and it helps to move the residue towards the large intestine so it can eventually be defecated. So segmentation is this is how you get segments of food that needs to be digested. And this is the movement where ring-like constrictions appear in several places along the intestine so that it's segmented off. And the rhythm of this is set by enteric pacemaker cells in the muscularis externa, which is the outermost layer of the small intestine. We talked about peristalsis, and peristalsis is the wave of the movement of the contents of the small intestine towards the large intestine, and this can occur in motions like the peristaltic wave. And when multiple successive peristaltic waves overlap, this is called the migrating motor complex. So we'll end here, and